Tooth structure and development. Each tooth is composed of three parts. Crown is a portion of the tooth that projects above the gingiva, gum. One or more roots are situated below the gingiva that hold the teeth in the bony sockets called alveoli, one for each tooth. A nick is narrow part between the crown and root. These are made up of three specialized tissues, but at the first you must know that all of tissues which compose tooth can be divided, depending on the structure, to hard tissues and soft tissues. And hard tissues, there are specialized tissues which are dentin, enamel, and cementum. Soft tissues, there are pulp and periodontal ligament. So within a tooth, there is a central pulp cavity occupied by dental pulp. Let's start from the dentin. The dentin is a calcified tissue of the tooth. Calcified, yes, is covered by enamel on the crown and cementum on the root and surrounds the antripulp. Dentin is made up of 70% inorganic materials. All of hard tissues contain inorganic component. So in general, they can be divided to organic and inorganic component. So let's come back to the dentin. Inorganic materials take 70% of volume of the dentin. 0-hydroxyapatite, which is a crystalline calcium phosphate. 20% organic materials, which is a collagen proteins, and 10% of the water. The organic matrix of dentin is secreted by odontoblasts. Odontoblast is here. Odontoblasts are cells that line the internal surface of the tooth, separating from the pulp cavity, so take peripheral place in the pulp. Odontoblasts have a structure of protein-secreting cells. These cells have branched cytoplasmic extension named odontoblast processes that penetrate perpendicularly through the dentin. Dentin consists of microscopic channels called dentinal tubules. Dentinal tubules, which radiate outward through the dentin from the pulp to the exterior cementum. From the internal part of the dentin to the external part, from inside to outside. And these dentinal tubules directed parallel to each other, directed radially. This tubule contains fluid and odontoblast processes. So bodies of odontoblast take place inside in the central pulp cavity and take peripheral part of the pulp, and they send long processes which are located inside intestinal tubules together with a little bit of fluid. There are three types of dentin. Primary dentin, it's the outermost layer of dentin, is body the enamel. The outer layer closest to enamel is known as the mantle dentin. This layer is unequal to the rest of primary dentin. Mantle dentin is formed by newly differentiated odontoblasts. Below it, the circumpalpal dentin is situated. This is more mineralized dentin, which makes up most of the dentin layer. Secondary dentin, regular secondary dentin, is a layer of dentin produced after the root of the tooth is completely formed. It grows much more slowly than primary dentin. It has a similar structure to primary dentin. Also, deposition is not always even around the pulp chamber. And tertiary dentin, irregular secondary dentin or reparative dentin, is created in response to stimulus such as a curious attack. So after the mating of previous dentin. Let's go to the next substance or next tissue, hard tissue, which is the enamel. The enamel along with dentin, cementum, and dental pus is the hardest substance of the human body. And this tissue is the richest in calcium and avascular. It consists of 95% of calcium salt, mainly hydroxyapatite, 1% organic minerals, collagen proteins, and 4% of the water. The enamel contains elongated enamel roots and inter-root enamel. Enamel root is a tightly packed mass of hydroxyapatite crystals in an organized pattern. Enamel rods are found in rows along the tooth, and within each row, the long axis of the enamel root is generally perpendicular to underlying dentin. Both interroot enamel and enamel roots are forms the hydroxyapatite crystals. They differ only in the orientation of the crystals. Let's go to the cementum. The cementum is specialized calcified substance covering the dentin of the neck and root of the tooth. Cementum is similar in composition to bone, and it's composed by the chemical structure of the cementum. It has 65% of inorganic materials, mainly zero hydroxyapatite, 23% organic materials, mainly collagen type 1, and 12% of the water. Distinguish two types of the cementum. Acellular cementum has no cellular components, covers all surface of the root of the tooth and try to be located near the apex of the root, near to the neck of the root, excuse me. And next type, cellular cementum, cover the lower part of the root, try to be located near the apex, and contain cells which are cementocytes. The main function of the cementum is take part in maintaining of the tooth. It's a holding of the tooth within the alveolar, dental alveolar, and the connective part attachment of the alveolar of the root of the tooth to alveolar wall, together with periodontal ligament. Next type tissue of the tooth, it's a soft tissue, which is a dental pulp. The dental pulp is the central part of the tooth, dental pulp, filled with a soft connective tissue. The central region of the coronal and auricular pulp contains large nerve trunks and blood vessels. And dental pulp has 3 layers. It's like zones. Cell-rich zone contain fibroblasts and undifferentiated mesenchymal cells. Cell-free zone rich in both capillaries and nerve network. And odontoblastic layer contains cell bodies of the odontoblast. So last zone, the peripheral zone, filling odontoblastic bodies. Cells found in the dental pulp include fibroblasts, odontoblast, macrophages, granulocytes, mast cells, and plasma cells. It's logically because the basic structure of the pulp is a loose connective tissue, and here can be present all of cells of the connective tissue together with capillaries, nerves, and lymphatic capillaries. Periodontal ligament. It's a second soft tissue which composes the tooth. It is composed of special type of the dense connective tissue whose fibers penetrate the cementum of the root and bind it to the bony wall of the alveolar. It was the information about structure. Let's go to the development. Tooth development. At about 6 weeks of gestation of the oral epithelium proliferates into the nivaginate into the interlining mesenchyme and forms the dental lamina. The dental lamina connects the developing tooth bud to the epithelial layer of the mouth. Tooth development is divided into the following stages: bud stage, cup stage, bell stage, and maturation or crown stage. Bud stage. In some places of the oral cavity, happen aggregation of ectodermal cells with formation group of cells, tooth bud. The tooth bud is the group of cells at the end of the dental lamina. Cup stage. Mesenchymal cells aggregate near the pole of the tooth bud. These cells are called the dental papilla. So happen evagination of these test buds, evagination by the dental papilla, which originates from the mesenchyme. At this point, the tooth bud grows around the mesenchymal aggregation, taking on the appearance of a cup. And because the enamel organ, a condensation of mesenchymal cells called the dental follicle, surrounds the enamel organ and then limits the dental papilla. So at the first happen formation, the enamel organ, and next stage, formation of the dental papilla, and the last, outside, happy organization of the dental follicle. Eventually, the enamel organ will produce enamel. The dental papilla will produce dentine and pulp, and the dental follicle will produce all the surrounding structures of a tooth. Bell stage. The dental organ is bell-shaped during this stage, so that it got the name bell stage. Cells of enamel organ are divided into 3 layers. Cuboidal cells on the periphery of the dental organ are known as the outer enamel epithelium. The columnar cells of the enamel organ adjacent to the dental papilla are known as inner enamel epithelium. The stellate reticulum is a group of cells located in the center of the enamel organ of the developing tooth. These cells are star-shaped and synthesize glucose aminoglycans. The cells between the inner enamel epithelium and the stellate reticulum form a layer known as a stratum intermedium. The cells of inner enamel epithelium give rise to enameloblast, which produce enamel. The dental papilla contains cells that develop into odontoblasts, which are dentine-forming cells. Mesenchymal cells within the dental papilla are responsible for formation of tooth pulp. The dental follicle gives rise to the cementoblast, osteoblast, and fibroblast. Cementoblasts form the cementum of a tooth. Osteoblast give rise to the alveolar bone around the roots of teeth, and fibroblasts develop to the periodontal ligament which connect teeth to the alveolar bone around cementum. And last stage is a maturation. Hard tissue, including enamel and dentine, develop during this stage of tooth development. Dentine formation is the first identifiable feature in the crown state of tooth development. Odontoblast, the dentine-forming cells, differentiate from cells of the dental papilla. They begin secretion and organic matrix, which contains collagen fibers around the area directly adjacent to the inner enamel epithelium. Further, odontoblast begins to move towards the center of the tooth, forming an excretion called the odontoblast extension, called the odontoblast processes. Amelogenesis is the formation of enamel on teeth and occurs during the crown stage of tooth development after dentinogenesis. Cementogenesis occurs later in the development of teeth. The cementoblasts differentiate from the cells of dental follicle. The cementoblasts secrete matrix of proteins and collagen fibers. Then mineralization takes place. So, finally, such type hard tissue as enamel origin from the ectoderm, and dentin and cementum from mesenchyme. Such types of tissue as a pulp and the periodontal ligament origin from the mesenchyme